to get going here this morning, I'd like to say thank you, first of all, to the sponsors and the meeting organizers for allowing me to present today, which we're going to be talking about our research on 9 years of selectively breeding tilapia for disease resistance. And the title of my talk is Where Are We and Where Are We Going? So just a little bit about me. I know I had a great introduction, a little more background here. I did my undergraduate at Iowa State University in animal ecology with an emphasis on aquaculture, after which I went to the University of Idaho and completed my master's as well as my PhD degree, where we're working with the rainbow trout industry on vaccine approaches for bacterial cold water disease or rainbow trout fry syndrome caused by Flavobacterium psychrophyllum. Once I completed my graduate work, I was fortunate enough to get hired in 2008 by the USDA-ARS here in Auburn, Alabama at the Aquatic Animal Health Research Unit. So I've been here for 15 years now, and am now also the lead scientist here. So a lot of our research, both in my laboratory as well as our unit here, is looking at vaccine development for warm water fish, looking at genetic diversity of pathogens, how do pathogens cause disease in fish, as well as co-infections, and as well as selective breeding, which I'll be talking more on today. So you may not be familiar with the USDA-ARS, but here in Alabama, we're the Aquatic Animal Health Research Unit. And again, our mission of our lab is to conduct applied research to solve fish health problems in aquaculture. So at our laboratory, we have 7 scientists with expertise in immunology, microbiology, parasitology, molecular biology, nutrition, and bioinformatics. And we work on a number of different fish species. Of course, we're working on tilapia, which we'll be talking about today. But we also do a large percentage of our research in channel and hybrid catfish, as well as a little bit work with largemouth bass, and more recently, some shrimp research as well. So with that background, just wanted to first introduce some important diseases of tilapia, which we're all aware of. Some of the important bacterial diseases impacting the global industry include Streptococcus eneae, Streptococcus agalactiae, Francisella orientalis. Culminaris disease can be an issue, especially in younger stages of tilapia aquaculture. These are caused by culminaris-causing bacteria, Flavobacterium aureochromis and F. davisi appear to be most predominant in the tilapia industry. There's Aeromonas species that can cause problems, as well as the Edwardsiellas, Ictaluri, Piscicida, Angularum, and Etarda at times as well. On the flip side, for viral diseases, there's a number of different viral diseases of tilapia of importance. The first being tilapia lake virus, as well as nervous necrosis virus, infectious spleen and kidney necrosis virus, as well as iridoviruses. But for today's talk, I'm going to primarily be focusing on streptococcal pathogens, Francisella, as well as tilapia lake virus. So the first pathogen I want to talk about briefly is Streptococcus eneae. This is a causative agent of streptococcal disease in tilapia. It's a gram-positive bacterium with a worldwide distribution. It forms or it has a hemolytic or beta hemolysis on blood agar. You can see here in this picture, we get the zone of clearing where it's lysed the red blood cells. And in the fish, a lot of times we see exophthalmia shown in this picture, as well as the C-shaped body formation, which results in spiral swimming behavior. And this pathogen can result in large mortalities under certain situations or conditions. Another causative agent of streptococcosis is Streptococcus agalactiae. Again, this is another gram-positive bacterium. In tilapia, there have been 5 different capsular types reported, that being 1A, 1B, 2, 3, as well as the others. And, probably of great concern in the current situation is the recent emergence of this 1A capsular type 2021 in Latin and South America, which is currently raising big problems for the industry. Similar signs of disease as strep enae with bilateral exophthalmia, as well as some strange swimming behaviors as well. The next bacterial pathogen we're going to talk about is Francisella orientalis. It's the causative agent of franciscellosis. And this is a gram-negative bacterium. It's also intracellular, so it can live within different host immune cells. And the mortality with this pathogen can be chronic at times. And some of the clinical signs of disease include skin ulcers and these really severe granulomas in the spleen, as well as the head kidney. So in the picture on the left-hand side, we have a normal spleen, where on the right, we see all these nasty granulomas within the spleen. And then the other picture kind of shows the spleen again with all these granulomas in it. So for viral pathogens, we'll talk about tilapia lake virus. We all know this is the causative agent of tilapia lake virus disease. This is an enveloped, a negative sense, single-stranded RNA virus with 10 segments in the genome. Some of the gross clinical signs of infection include skin lesions, as shown in the middle picture here. Scale protrusion is quite commonly observed, which is shown in the right picture, as well as some fairly consistent brain and liver pathology associated with this virus. And another feature of this virus is co-infections appear to be very common with this virus. So we have these different diseases, and we know there are different methods to try to prevent and control them. Usually, one of the first steps is biosecurity, as was mentioned in the previous talk. Different management strategies, such as stocking at lower densities. There are some vaccines available. With the bacterial pathogens, some antibiotics are available as well for use. There's probiotics, organic acids, and essential oils being included into feeds. And all of these can be effective, and they're all part of a good health management program. But even with these in place, economic losses can occur. So a complementary approach to these other disease prevention and control options includes selective breeding. We know from Atlantic salmon, which breeding has been going on for much longer than in tilapia, that as well as other fish species, the resistance to disease in fish can be heritable, depending on the fish species, as well as the pathogen. And when we started working on this, very little was known or had been done in terms of selectively breeding for disease resistance in tilapia. So we formed a partnership in 2014 to begin looking at the ability to selectively breed for disease resistance, because at that time it really was not known. So we had teamed up with Spring Genetics due to their great expertise in tilapia breeding and production. And the other partners were the Akavorsk Genetics Center, which is now Benchmark Genetics. And they have a strong expertise in quantitative genetics and leading numerous selective breeding programs across the world. And then at our laboratory here at the Aquatic Animal Health Research Unit, we had expertise in diseases of tilapia. So we formed this team to tackle this problem. I just wanted to introduce some of the players in this team. So in this picture here, you can see our location here in Alabama and Spring Genetics. So we're only about 12 hours apart, which allows us the ability to transport fish from Spring Genetics up into our laboratory for research. So some of the players at our laboratory, myself, as well as Dr. Craig Shoemaker and Dr. Ben Beck. At Spring Genetics, some of the leaders there include Jose Espinorenga, Carlos Lopez, as well as Heidi Yoshi, Segovia Uno. And then at Benchmark Genetics, led by Dr. Morton Rye, Carlos Lozano, as well as Sergio Villavicuo. So this is a team that we assembled in 2014 to begin to look at the ability to selectively breed for disease resistance. So the next part of my talk, I'm going to be talking about where are we. We've been breeding now for 9 years, and I just want to give a current status of where we're at for selectively breeding tilapia for resistance to Strep Streptococcus agalactiae, the capsular type 1b, Francisella orientalis, tilapia lake virus, and then talk a little bit about the use of genomic information for enhancing selective breeding for resistance. So when we started this collaboration, our objective was really to selectively breed for disease resistance to these pathogens. So we conducted a large amount of research to answer some important questions, including, do some families of tilapia survive better than others? Or is there genetic variability in resistance to disease? If there is, can this trait be passed on to offspring? And then also, are there any individual genetic markers or DNA markers that are strongly linked to the expression of disease resistance? So the way our collaboration has worked is down at Spring Genetics in Miami, Florida. They've been breeding for a number of different generations. So when we started in 2014, that was the third generation. We're now up to the 12th generation, the Spring Genetics breeding program. So every year, Spring Genetics breeds their families of tilapia for a number of different traits, including growth, resistance to Strep, resistance to Strep agalactiae, Francisella orientalis, and now tilapia lake virus. So every year they generate or produce about 145 different families of tilapia, which those families are individually reared as eggs until they hatch into fry, until they're large enough to be pit tagged. So once they're large enough to be pit tagged, they're inserted with a little tag that we can know exactly who that fish is and what family it came from. And at the same time, those fish are being tagged, a piece of fin tissue is taken for genetic analysis, as shown in this figure, for looking for these signatures of disease resistance. So after all the families have been tagged, they can be all placed together and no longer be held as separate families, at which time those fish are netted and transported by truck to our laboratory here in Alabama to do pathogen challenges. And here's just a few pictures of us receiving fish over the years. So once we get the fish at our unit, the goal is to, whichever pathogen we're working on in that study, is to infect all those fish with the pathogen of interest, and then look at who dies and use this data for selective breeding. So in essence, for the methods are first to grow the pathogen. So in the top left-hand side, we see a tissue culture flask for growing tilapia lake virus. For the other bacterial pathogens, we grow them on plates and then in broth. At which point, for the challenges, we are using injection challenge models for all four of these pathogens, in which the Strep agalactiae and Francisella are being injected intramuscularly, versus Strep and tilapia lake virus being injected in the coelomic cavity or intraperitoneal cavity. So after we infect them with the pathogen, they're all placed into a single tank. And the numbers of fish, again, are 145 families on average and about 20 fish per family. So all these fish are infected and placed in the same tank, after which we monitor them for disease signs and mortality. Once we do find a fish mortality, they're netted out of the tank. And then we perform a necropsy for several different purposes. Firstly, to scan the fish to get that pit tag to understand who this fish was and what family it came from. Then we also take a portion of the brain to re-isolate the pathogen they're infected with to confirm that that was the cause of death. So in the cases of bacteria, we put those on bacteriological plates, versus tilapia lake virus, where we add homogenates of the brain onto a cell line conducive for the virus to grow and divide. So we can look for cytopathic effect to confirm that those fish had died due to the virus. And again, here on the right are just a few different pictures of us in the process of challenging these large fish. So each family challenge has about 2,800 fish. So how is this data used? And I'm just going to go over for each of these pathogens what we've done over the years. So in this first figure on the left is a graph showing the cumulative mortality over time after fish from the third, fourth, and fifth generation, or excuse me, sixth generation were challenged with Streptococcus eneae. So this is our general challenge model with Streptococcus eneae. It's a fairly rapid disease progression where most of the mortalities occur about 1 to 3 days post-challenge, after which mortality is pretty much over. So in each of these challenges, we average anywhere from 30 to 60% mortality. However, this is the overall from all fish in that study. But if we look at who died within these fish, we see this picture shown on the right, where we have families of fish on the x-axis and survival, in this case, on the y-axis. What we see here is we have some families with very poor survival at 0%, all the way up to families that had very great survival at 100%. So when we share this data with a quantitative geneticist, they run different models to look at whether this trait is heritable. Our data has indicated that yes, our heritable estimates for Strep eneae are quite high, ranging from 0.42 to 0.52, depending on the generation and the challenge, which suggests that increased resistance should be able to be passed on to offspring. So in order to confirm that, the following year, in addition to making the 145 families, within that, several families are created by crossing resistant fish together, in which if this trait is truly heritable, those progeny should be resistant to disease. Then a few families of susceptible fish are also crossed together, in which their offspring should be more susceptible to disease. We take those offspring, infect them with Strep eneae, and then look at what levels of survival are obtained in the fry. And what we see is shown in this bar chart on the right with survival on the y-axis and the average family compared to the resistant and the susceptible parents. What we see is when we cross resistant families together, we get much better survival than the average family. And when we cross susceptible fish together, we get very poor survival as expected, showing that resistance to Streptococcus eneae is heritable, and we can improve this trait through selective breeding. So next, I'm going to move on to our work with Streptococcus agalactiae. And again, currently, we're working with the capsular type 1B, because at the time when we started this research, that was the most predominant capsular type impacting the industry. So again, very similar data shown here on the left graph, left graph. We have day post challenge on the x-axis and cumulative mortality on the y-axis. You can see our challenges from the 4th generation all the way through the 11th generation. So we've done a number of challenges over the years with Streptococcus agalactiae. And again, our challenge models are variable. It's never perfect. We don't always get the mortality we shoot for, but we've been pretty effective in a reproducible model to get anywhere from 30 to 70% mortality each generation. And we would look at within this overall mortality who died, similar to Strep eneae, we see we have some families with very good survival on the far right-hand graph, where we have families on the x-axis and survival on the y-axis. And we see here again that we have some families that perform very poorly with 0% survival. However, on the other spectrum, we have families with very high survival. And again, when we share this data with the quantitative geneticist and they run this data through their models, our heritable estimates have been quite high, around 0.4, suggesting that, again, increased resistance should be able to be passed on to offspring. So when we've gone to confirm this, again, very like the Strep eneae research, where we cross families with very poor survival that should be susceptible to disease, we're crossing families with very high survival that should be resistant to disease. We grow those progeny up with the next generation, challenge them with Streptococcus agalactiae. And again, we found that resistance to Strep agalactiae is also a heritable trait. So in this mortality graph on the right, we see the average family had about 55% mortality, where those fish bred from susceptible parents had very poor survival, at almost zero survival. However, those fish that were crossed from resistant parents, their offspring were very resistant to disease, with survivals greater than 80%. So what this data showed is that, again, with this pathogen, it's a heritable trait, and we can improve it through selective breeding. So next, I'll talk about the Francisella orientalis research. Again, you're going to see the very same graphs. On the left-hand side, we have days post-challenge on the x-axis and cumulative mortality on the y-axis, where we've done we've challenged 4 generations of fish with Francisella. Again, our mortality is ranged in these challenges from, we had one really low survival or mortality challenge at about 10%. However, most of them have been from 20 to 50% mortality, which has been enough data when we look at who's dying within that study. Again, we have this nice spectrum of families ranging from those with very poor survival to families with very high survival. And when this data is ran through models for quantitative geneticists, the heritability estimates have been around 0.3, which again suggests that increased resistance to offspring should be a heritable trait. So again, we wanted to confirm this. So over the generations we've done this research, we've crossed families that are very resistant or had very good survival to challenge. So their offspring should be resistant. Those were crossed, or excuse me, then we also had crosses of families that performed very poorly against in survival to Francisella orientalis. So their offspring should be susceptible. So when we challenge those offspring with Francisella orientalis, what we see is that the average family in this particular example here in the mortality graph on the right, the average family had about 50% mortality. However, those progeny that came from resistant families had very high survival, around 80%. And those families, or those progeny that came from families that were susceptible to disease, their offspring were very susceptible to disease. So again, with this pathogen, we're also able to demonstrate that we can improve resistance to Franciscellosis through selective breeding. So some of the newest research that we've been conducting is looking at the ability to selectively breed for resistance to tilapia lake virus. So we've just recently conducted this research within the past year. So we've only done one challenge so far with the 11th generation. And here's our mortality on the left-hand figure with day post challenge on the x-axis. And again, cumulative percent mortality on the y-axis. Again, using our challenge model, we got a very good disease pattern over time, which resulted in about 70% mortality over all 145 families in that study. But again, on the right, when we look at who's dying within that, what we see is we have a number of families with very poor survival at 0%. However, on the other spectrum, on the right-hand side of this graph, we have families that perform very well against tilapia lake virus with survival above 80, 90, almost near 100% survival. So again, when this data is ran through quantitative genetic models, we find that the heritability is moderate at about 0.2. We see that some families are performing better than others. And this data shows that resistance to tilapia lake virus is a heritable trait. And we have not done this research yet, but we will be soon testing to confirm that in studies upcoming. So in terms of where are we, what we've shown through our research is that it's possible to selectively breed Nile tilapia for resistance to Streptococcus eneae, Strep A galactiae 1b, Francisella orientalis, and now tilapia lake virus as well. So in the big picture, in the spring genetics tilapia, these fish have been bred for resistance to Streptococcus eneae for 3 generations based on phenotypes. However, a number of different generations via marker-assisted selection, which I'll talk about here in a second. For Strep A galactiae 1b, we've been breeding for 8 generations now, in which we're utilizing genomic selection. For Frentz orientalis, we've been breeding now for 4 generations, again, utilizing genomic selection, which I'll talk about as well. And now for tilapia lake virus, which is the newest pathogen online, the 12th generation now that is being produced right now, has a breeding component for resistance to tilapia lake virus as well. So we know that we can breed for resistance, but we're also interested at, are there any genetic markers that will strongly correlate with this resistant phenotype? So with benchmark genetics, we've been pursuing this research. And just going back to my previous figure, we're going to talk a little bit more here about the fin clips, which we've extracted DNA from each individual fish going into every study I showed, and then the genetic signatures for resistance. So first, I'm going to talk about our work on Streptococcus enyei. So we performed a genome-wide association study. So, basically, all this study did was look at the DNA of families of fish that were either susceptible to disease or very resistant to disease, and looking for any DNA marker that is unique to those fish that were resistant to disease. And what we found through this genome-wide association study is we identified several genetic markers that were strongly correlated with resistance. So on this figure here is just showing different chromosomes or linkage groups of the tilapia genome. And then here in this linkage group 8, we see all these markers above this red line. And these were the genetic markers that correlated strongly with resistance. So this suggests is that we may be able to breed for resistance based on the DNA and not have to do all the pathogen challenge work, but we wanted to confirm that that is possible. So in order to confirm that we can breed for resistance based on this genetic marker, the folks at Spring Genetics, they looked at families that have that DNA marker, as shown in this top picture here. So families with that marker were bred together, or fish with the marker were bred together. And those I'm calling good fish, which should be resistant to disease. Then they also created progeny from families that did not have the marker. We're going to call those bad fish, and they should be susceptible to disease, to Strep enyei, if we can breed on this marker. So we obtained 300 fish that should be good and 300 that should be bad. And then we infected them with Strep enyei to see what mortality happened in those individual fish. So when we did that, again, a similar figure here on the left, we have the day of the challenge on the x-axis and cumulative percent mortality on the y-axis. In this particular challenge of all 600 fish, we had about 40% mortality overall. However, if we look at which fish were dying in that study, here on the figure on the right is a survival probability curve or survival curve. And what we see is those fish bred without the marker for resistance performed very poorly with very poor survival. However, those fish that were bred to be resistant based solely on that genetic marker had outstanding survival, where I think we lost only 1 fish due to disease. So what this research showed us is that offspring produced from parents with the genetic marker were resistant to Strep enyei. So now that we have unraveled that research, they're now at Spring Genetics are using marker-assisted selection for breeding resistance to Strep enyei. So in terms of the other pathogens for Strep A galactiae 1b and Frentz orientalis, we have not identified any major QTLs that strongly correlate with resistance to those pathogens. However, we are utilizing genomic selection to enhance our ability to selectively breed for these pathogens. So genomic selection uses genomic information from the families, from a very large number of markers with a small individual effect across the tilapia genome. So in essence, we can utilize some genomic information, combine that with the phenotypic data, or how well they survive in laboratory challenges to disease. And by using this genomic information along with their phenotype, we can increase our ability for accurate selection for disease resistance against these pathogens. For tilapia lake virus, this research is underway, and we don't have any up-to-date results on that as of yet. So the first part of my talk is where are we? And now I'd like to take a few moments to talk about where are we going in our selective breeding program. So I'm going to spend just a few moments. One component that we're interested in is we know that we can breed for resistance. What we don't know is why are they resistant? What's the underlying mechanism for disease resistance? Basically, how do these resistant fish fight infection? And then we're also looking ahead for selective breeding for new pathogens, which is driven by the needs of the industry. And we'll use an example being a strep A galactiae 1A 2021 that has recently emerged. So we're interested in research on how are these fish resistant. And we believe that understanding the mechanisms for disease resistance is going to be important in just basic knowledge of what we're breeding for. But we believe this knowledge can also be used to enhance our breeding as well. So in terms of the Streptococcus eneae, I talked about the good fish and the bad fish, and this phenotype is absolutely phenomenal in that when we do laboratory challenges with these fish, the good fish overnight are very healthy. They have no signs of disease. They'll eat whatever we feed them, and they perform very well. However, the bad fish, they perform very poorly. They have mortality overnight, nearly 100%. So we have a really good phenotype that we can study to understand the mechanism behind this. So some of our current research is, looking at how do they fight off the infection. So this figure here on the left, where we see a time course from 0 hours to 24 hours with susceptible on the left-hand Petri dishes and resistant on the right-hand side, where we infected both resistant and susceptible fish with Strep eneae. Then we took a sample of the brain and plated it onto sheep blood agar and just looked at what loads of bacteria are in the brain of these fish over the course of 24 hours after being infected. What we see here, we can compare what's going on. In the susceptible and resistant fish at 0 hours, there are no bacteria as expected. However, at 2 hours, we start seeing bacterial colonies. And again, these are beta hemolytic. So what we're looking at are colonies of Strep eneae with a zone of clearing where they've lysed the red blood cells in these pictures. So we see at 2 hours, we have some bacteria starting to build up in the susceptible. We also have some bacteria in the resistant fish as well. However, as time progresses, at 4 hours, we see this infection building up in the brain of susceptible fish. However, in the resistant fish, that infection never builds up. We do pick up some bacteria in the brain, however, at very dramatically reduced loads to the susceptible fish. So we want to know what's driving this. We've recently done some pathology work on the fish where resistant and susceptible fish have been infected with Strep eneae, and then looking at histopathology for what pathological changes may be going on in these fish. And what we found is pretty interesting in that in the susceptible fish, as shown in these two pictures here on the right, where I'm showing some inflammation in the spleen. So in this box here on figure A, We see some inflammation of the spleen of susceptible fish. If we zoom in on that box on figure B, what we can see are inflammatory cells in that region, as well as a large amount of coccoid or Streptococcus eneae bacteria. So we want to know, but yet, in the resistant fish, we don't see hardly any Streptococcus bacteria and none of this pathology, or very mild pathology. So in addition to the histopathology, we're looking at molecular approaches to look at the immune responses in these fish. So we're doing some RNA research and what genes are being differentially expressed between the resistant and the susceptible fish after being infected with the bacteria. And we hope to unravel our understanding of why these fish are resistant, and believe that this will help just our basic knowledge of what we're breeding for, but perhaps for assisting in improving our breeding program as well. So the second part of where are we going is always keeping our eye on the industry needs and looking at emergent pathogens. So as many or most of you know on this meeting today, that there's been an emergence of the Strep A galactiae capsular type 1A, being called 1A 2021. We know that this variant of Strep A galactiae, the disease is presenting itself quite differently than the traditional Strep A galactiae for 1B, where 1B, typically the disease was in 150 to 300 gram fish is where we'd see streptococcosis. But with this new strain, we're seeing a very diverse background of sizes of fish being affected. I did not say that well at all, but we're seeing this disease in very young fry all the way to broodstock. So the pathology between the 1B and the 1A is quite different. And one thing that we want to know is we've been breeding for resistance to the strep 1B, but obviously we're very interested in knowing what is our breeding done for resistance to this new 1A 2021. So we're beginning to gear up for that research this year. The first thing we needed was a industry relevant isolate of this new Strep A galactiae 1A 2021. And we've recently teamed up with Dr. Paula Barato. She assisted our lab in obtaining one of these new isolates from Regal Springs in Honduras from Miguel Madrid. So I appreciate the working together on this so that we could get an isolate to our laboratory here. So now we're starting to begin to look at developing challenge models in the lab. They're going to mimic what's seen in the industry. So we're currently right now testing different laboratory challenge models using either intramuscular or intraperitoneal injection. We're also probably going to look at immersion challenges as well. And again, the goal of this is to get a challenge model that we can reproduce and use for a larger study with all families of fish. And that's exactly what we intend to do. So we are gearing up with the 12th generation to challenge those families with Strep A galactiae 1A. And again, answering some of those same questions that I went over with the Strep eneae, Strep A galactiae 1B, and Francisella and tilapia lake virus. But we want to know, is there genetic variability and resistance? Has the breeding we have done, is it also effective for this 1A? If there is genetic variability and resistance, we want to calculate those heritability estimates and then incorporate selective breeding for resistance to this new variant in the selective breeding program. So this is very current research and we're just getting going on it right now. So in conclusion, our research over the last 9 years has shown that it is possible to selectively breed tilapia for resistance to Strep eneae, Strep A galactiae 1B, Francisella, as well as tilapia lake virus. In terms of Strep eneae, we have a major QTL that correlates with resistance. And one thing I did not talk about, but we've looked into, there are no negative effects of breeding on growth or any correlations between resistance to the 4 pathogens. So if we breed for Strep EE, that doesn't improve or doesn't make fish any worse for growth or Strep A-galactate. It just means that we have to breed for all these traits together. And just finally, we talked about golden bullets in the last talk. It's silver bullet here in my talk, but using disease-tolerant strains of tilapia is just another tool in our toolbox that we can use for trying to reduce disease-associated losses in tilapia production. We have vaccines, biosecurity, antibiotics. We have all these other tools, management as well. But another component can be the genetics of the fish that are using. It's not a silver bullet, but again, it's another tool in the toolbox for trying to improve our efficiency and sustainability of tilapia aquaculture. So just finally, I'd like to acknowledge some funding sources for this work, as well as a number of people who have helped with this research. Our research has been given some awards over the last year or two with the impact of the research has had on the tilapia industry. And finally, if you're interested in contacting me after this meeting, here's my contact information through my email, LinkedIn, or Twitter feed as well. So I don't know if I have time or not. I think I got us back on time, but I'd be happy to try to entertain any questions you might have.